live வரும் 5 seconds கழிச்சு பேச ஆரம்பிங்க மேம்.
Yes. Good evening. I welcome you all to the final day of the week-long alumni lecture series on opportunities in biosciences, organized by the Department of Biochemistry, Microbiology, and Postgraduate Department of Bioinformatics and Applied Microbiology. Our erudite alumni from the Department of Biochemistry and Microbiology spoke on the various avenues in life science. It was a rich learning experience for each and every participant. Today we have with us Dr. Sahaja and Dr. Masai Deepika, our alumni from the Department of Biochemistry, to share their knowledge and research experience. I'm sure most students will be inclined to pursue research as it has so much of scope and highly challenging. I would like to thank our correspondent Dr. Lata Rajendran and Chairman Mr. Kumar Rajendran for their constant motivation and support. I would like to congratulate each and every faculty of the Biochemistry and Microbiology Department for organizing this webinar and for learning how to use this online platform to disseminate knowledge. Thanks to all the participants who have responded to our invite and participated in this webinar. Over to Dr. Victoria, Head Department of Biochemistry, for the session.
Good evening. We have been acquiring knowledge on various topics given by the alumni students of the departments of Biochemistry, Microbiology, and Bioinformatics. Today, we have another skilled and hardworking student, Dr. Sahaja Aigal. Dr. Sahaja Aigal is currently a postdoctoral researcher at the Institute of Medical Microbiology, Virology, and Hygiene, University Clinic Hamburg, UKE, Germany. She has completed her undergraduation from the Department of Biochemistry, Dr. MGR Janaki College of Arts and Science for Women, and her post-graduation from the Department of Biotechnology, University of Madras. She was awarded the International Max-Planck Research School Fellowship to pursue her research career at the Max-Planck Institute of Immunobiology and Epigenetics, Freiburg, Germany. Her area of interest include host pathogen interaction studies, super resolution, optical imaging, drug delivery methods, and the field nanotechnology in health and medicine. She is currently working on visualization of Yersinia enterocolitica infection niche in mammalian host cells. Dr. Sahaja Aigal has to her credit reputed patents and numerous publications in esteemed journals. We are happy to have such an ambitious student to take over the session. Over to you, Dr. Sahaja.
Thank you, ma'am. Thanks for that very kind introduction. So good evening, everyone. Today, I would be telling you about some study and research opportunities in Germany. But before going to that, I would also take you down memory lane to the time when I started doing my bachelor's in biochemistry and how I went on to do infection research and so on. And then, of course, I would talk about the higher studies in Germany. So my journey so far, I studied in MGR Janaki College for BSc Biochemistry. And there it was very fascinating to see several different topics of biology that interested me. And I had very good knowledge foundation of knowledge was laid by very good lecturers in the college in biochemistry and microbiology department. And so the bachelor's was basically to gain a lot of theoretical knowledge, and that laid the foundation for my higher studies. And then I did my master's in University of Madras Guindy campus, Department of Biotechnology. And there again, the theoretical understanding further deepened. And apart from that, I also went to do some internships and gain some hands-on experience in research. And I also did master's thesis in CLRI, and I got a small publication from there. It was the thesis was based on drug delivery. And then I took 2 years. I worked as a project associate in IIT Madras. So it was a department of chemistry, and under Professor Pradeep, who is a recipient of Padma Shri this year. And it was a department of chemistry doing nanotechnology research. And I was asked to test the antimicrobial activity of nanomaterials for water purification. So you see, it was a very multidisciplinary project. And there I got to visualize the bacteria and stuff like that. And there was extensive research happening, and there was as a result of the work from the entire group, we got a publication which was published in PNAS. And here is an example of an image where the quality of the nanomaterials that were produced was tested. This was one of the ways, using microscopy. So basically, in water purification, we used nanomaterials which had nanoparticles within them, and they were supposed to release the only the ions. So silver nanoparticles were embedded within this nanocomposite, and they were supposed to release only the silver ions and not the nanoparticles when you're into the drinking water. And the silver ion release should also be in admissible standards. So we were just testing this. So this is how a normal bacteria would look under a dark field microscope. And then when you use pure nanoparticles without any protection, like no protection of any cover, and then they are just there, then they are into every bacteria, like you see it here in the hyperspectral image. You have different colors based on the size of the nanoparticle. It scatters in different light range. And then when you have the nanomaterial that is only releasing ions and not nanoparticles in the solution, and it does it has the ability to kill the bacteria. You can see the counter of the bacteria, and it's being and the white circle regions are the places where the counter is disrupted. So this effect was visible, and this kind of proved that the material was releasing only ions and not nanoparticles, which would be toxic. So like this, we did several such research. We did batch studies, and we were able to produce water cartridges. And it was also a startup company which is doing quite well right now. And so I gained a lot of practical experience from this group. And with this experience, I started applying for PhD positions. So I started first looking around within India, but I couldn't clear the NET exam for some reason. Probably I was jinxed. And then I thought, okay, if not NET, what else can I do? So I started looking for opportunities abroad. And I tried first with the US, but the applications were quite expensive for me. Back then, I didn't have good financial status. Then I thought, okay, let me try to apply in Germany. Why not? Because they have so many world-class institutes. And then I started gathering information about Germany, started to search in different PhD application portals, going to Nature Jobs website and so on, and looking at all the openings there. And then I also, at the same time, I cleared TOEFL and GRE and started gathering my transcripts, CV, and so on. And it was all online applications. So Germany did not cost anything to apply for a PhD position in Germany. So that was the best thing that I liked. That was why I actually chose Germany. And then I gathered everything, I applied online, and then I kept waiting. And then finally, one day, I got this selected for interview email. And then I applied for visa interview. And finally, I could fly. They also sponsored the flight and the stay there. There was an interview week in Max-Planck Institute for Immunobiology and Epigenetics. So it was an international Max-Planck Research School for molecular cell biology. So they had invited people from different countries. So we were a lot of people who were selected. And then for the interviews, and the interview week was quite intense because it started off with an oral presentation of the research that we did during our master's thesis. And afterwards, there was a panel interview with a lot of group leaders. And before this panel interview, they also gave us some research papers to read. And this panel interview kind of tested our ability to interpret the data which is in the paper. So they asked questions based on the research paper, which was totally not the topic that I was working on. And so this was a way to understand or to estimate our capability, understanding capability. And afterwards, if we somehow managed to impress the panel interview, the panel, then we were going to the next level, that is the one-to-one interview with individual professors. And I was lucky that I got through that stage. And then in the one-to-one interview, it's usually you get interviewed by the professor whom you feel might be a potential supervisor. So you have to give names of 3 people. And then it was 3 potential supervisors, one-on-one interview where they test your knowledge. And it is more like a conversation about your research aspirations and so on. After this, there was also a student panel interview where you talk to the students who are in their second year, third year, or final year students. So it was interesting also because this is a very important aspect. People usually underestimate it, but it's the student panel interview was mainly to understand how social you are. So other than research, what else? What other hobbies do you have? What kind of a social life do you like, and are you a friendly person? So this is kind of a thing that they look upon, and this is very important when you work in a research environment, because you need to contribute to the work environment, and you need to be a pleasant person also. And so this is something that they kind of understand. And so after this student panel interview, there is a discussion happening between the PIs and the students, and then finally they told us who is going to the next level, and I was also selected for that level, and that was the time for lab rotations and orientation week. So that is like kind of almost we are going to start the PhD project. So I was very excited. I went back to India, tried to prepare something, and after 3 months I came back to Germany, and we started off with the orientation week, wherein they talk about they give us orientation about different techniques that could be used in your research, and they also gave some general topics about time management, and then also about the facilities present there, the animal facility, the FACS facility, the microscopy facility, and so on. And afterwards, also then I had to do lab rotations, where I chose the 3 PIs of my choice, and then so each month, so 1 month was dedicated for 1 lab. So you have to work, you might have, I was given a mini project in every lab, and this way you get to understand how good you are, like where do you stand, and how much more you can learn. And it's also a way to understand how the PI would behave, and it's a thing that makes you understand where you stand and in research and what you like. So this was a best way to explore different labs, and then you choose the lab that you like the best. And I finally chose my lab, and that's how my PhD actually started after 3 months of rotation. Then I entered into the field of infection research. So as I said, during my master's thesis, I had a little bit of handling of the microscope during the drug release studies, but then I got much more insight into microscopy when I was doing my project at IIT Madras. And then that was the first time that I saw live bacteria swimming around, and that was quite fascinating. And in my PhD project, they said I'm going to work with bacteria again, but I will also look at the host pathogen interaction, and I can see this live. And this was something that really caught my attention, and I really wanted to do this PhD. And of course, that was my sole reason back then. And now, 10 years later, you see that we are with a pandemic, and now I think we can be much more appreciative of the infection research because so many people have died over so many centuries. The Toefl, for example, this is a bubonic plague that happened like several centuries ago, and it wiped out up to 30 to 50% of the population in Europe. It was caused by Yersinia pestis. And although this pandemic is gone, but there are still sporadic cases of Yersinia pestis infection even until 2020. In Africa and other places. So, but then you also have ongoing pandemics like the HIV and of course the coronavirus right now. So this kind of shows us that we are far from understanding all the pandemics, all the infections. And the major challenge that we have is that there is all these bacteria, all these pathogens are able to mutate so nicely, and they're able to, like for example, bacteria are able to become antibiotic resistant. And this is a reason why we need novel methods to understand them, and we should understand the mechanism even better. And that's where basic research is quite helpful. When you know the mode of action, the proteins involved in its interaction, you can get some idea as to what to target to get better drugs, or for vaccine research and diagnosis and everything else. So now coming back to my PhD project, I worked on Pseudomonas aeruginosa, which is a gram-negative opportunistic pathogen. So if you're healthy person, nothing happens, you're fine, your body's immunity will take care of you. But then if you're immunocompromised, it can lead to any kind of infection, right from the brain to the toe. You can have septicemia, you can have, like if you have burns, you can get sepsis of the burns, and you have something called swimmer's disease. And there are so many different conditions that the Pseudomonas can cause. And then, so this project involved the elucidation of the host cell membrane associated interaction partners of Pseudomonas aeruginosa and its lectins. And that means in space and time. So Pseudomonas aeruginosa, since it's a pathogen, it produces a lot of virulence factors. So lectins are some proteins which like to bind to carbohydrates, that is sugars. So since also human cells have a lot of sugar coating around them, it's a good way to go and bind to host cells. So during my PhD, the lab that I was working in, we were majorly focusing on the Pseudomonas aeruginosa lectins. And during this time, I learned several techniques. And PhD is a time of not just publishing, but more than publishing, what is important is learning. And I learned several things there, several techniques. So there was mammalian and bacterial cell culture, confocal microscopy, and then super resolution microscopy, and trying to analyze the images, bacterial invasion assay, and then normal basic techniques of Western blotting, pull-down assays, co-immunoprecipitation, and so on. And then this also led to a few publications. My major results of my PhD are not yet published, but these are some collaborative projects which were published. And this was the very first publication. Just when I entered the lab, I was involved in this collaboration. And here we saw that if the lectin, one of the lectins, Lec A, is deleted from the bacteria, it reduced the ability of the bacteria to invade cells by more than 50%. And also one more thing is this picture shows that this is actually a bacteria in green, and the strain that we worked on was PA01 strain. And when it's tagged with a fluorophore GFP, you can actually visualize this. And we have a synthetic membrane. It's called giant unilamellar vesicle. So it's kind of a mimic for host membrane, which is made up of lipids. And so it is known that the Lec A, the bacterial lectin, is able to bind to a specific receptor, which is a glycolipid, GB3, on the host cells. So what was done in this experiment was just they took a minimalistic system, so just a GUV, which had in it a marker for just to know the GUV is present there. So it's a Texas red stained phosphatidylethanolamin. And then there was GB3 added to this. And then it was made to, and the bacteria was made to infect this. And you see that it's able to form this kind of an invagination, which is the first step for invasion or endocytosis of the bacteria into the host cell, for instance. And so we said for this first step, there is no need for actin or any other cellular cellular components. It is just the receptor. So the binding of the receptor to the bacteria alone was enough for triggering the entry of the bacteria into the host. So coming back to infection research, we had so there are several techniques that you could use that are currently being used, but the major techniques are involving microscopy, because seeing is believing. And we have several different types of microscopy. We have the light microscopy, starting from basic wide field, confocal imaging, and then super resolution microscopy like DSTORM, PALM, and STED. And also we have cryo-EM, which is for so it goes even further down in resolution. It's so important in research, and that's why these techniques were also awarded Nobel Prize in 2014 for chemistry. Stefan Hell was given Nobel Prize, and Joachim Frank and colleagues were given Nobel Prize in 2017 for chemistry. Apart from this, you have also other techniques, of course, the mass spectrometry to understand the interaction partners, to deduce the host interaction partners, the interaction partners within the virus or within the bacteria, and then the gene editing techniques. And of course, you have a whole new area of bioinformatics. And here is just an image of electron microscopy image of coronavirus infecting viro cells. This is taken from the Robert Koch Institute website. So it's just trying to infect, and there are already some viruses inside. So this is an advancement of the microscopy technique that allows us to see how infection looks like in cells. And then also here you have the confocal imaging of nuclei of the lamin. And then these are the tubulins, and these are the cladherins. And also already in confocal, you see quite some detail, but you go down with resolution, the detail gets much more interesting. You have much more like separate spots, and you can get much more information. And this is a confocal image of nuclear pore complex. You see nothing at all with a confocal. And when you use a different super resolution technique, you see that there are two different layers, and you have this nice nuclear pore complex structures. So the reason why I actually showed this slide is that you see that when we talk about biochemistry or microbiology, it's not just about one area. Research is multidisciplinary. So for example, Stefan Hell won the Nobel Prize in chemistry for a technique that uses physics, a lot of physical property, because it's something to do with lasers, with the hardware of the microscope is all something to do with physics and engineering. And then it's used to image biological samples. So you see the connection, it's all so much interrelated. And that's why we need to really have an holistic approach to research, to science. Now, coming on to Germany, like there are a lot of institutions that are specializing in infection research. So I'm just listing a few of those, and the research foci, they of course do much more research than what I have shown here, but it's just to give you an overview. So you have the Robert Koch Institute, which is like the ICMR in India, for instance. So it's a public health hospital in Germany. It deals with a lot of epidemiological data. Like for example, recently for the coronavirus, it gives you the updates and so on. And then there is the Helmholtz Center for Infection Research, which deals with immune responses, anti-infectives, and so on. And then there's a German Center for Infection Research, which works with HIV, gastrointestinal infections. Then there's the Heinrich Petter Institute, which is mainly for virology. And then there is also an Institute for Tropical Medicine, where they also work on malaria and some less known or neglected tropical diseases. Then you have the Research Central Borsta, which works on mycobacteria. They also have a National Center for Mycobacteria. And they also work in interdisciplinary ways like immunobiophysics and also on asthma and allergy. Then you have the Max-Planck Institute for Infection Biology, which is dealing with host microbe interaction. They also use techniques ranging from genetics to cell biology, evolutionary biology, and so on. And then we have a quite recent add-on, that is the Center for Structural Systems Biology, where they look for structural biology of bacteria, of infection of bacteria, viruses, and so on. So they majorly use microscopy techniques. And then you have the Charité Institute, University of Medicine in Berlin, where they work on microbiome, mucosal immunology, etc. So why would you come and do research in Germany? So it was published in Nature recently that it's the golden time to do research in Germany, because the government is actually giving very good funding for research. So that's the major reason why people come to Germany for doing research. If you look at the number of researchers in European Union, Germany has the highest number of researchers working in science and technology and academia in the European Union in comparison to all the other major European countries. So basically it has excellent infrastructure, and the PhD students here are employees. They are not just students. So we have work contracts. So initially when I started off my PhD, we were given scholarships, and then it changed to work contract. So what is the advantage of that? That means the advantage is that it is considered as a job. So you have work experience. So you can apply, if you want to go out of academia, you can apply in industries saying that you have a work experience, and it's not a student status. Another thing is that you get covered for health insurance, and there's a very good pension scheme and the unemployment benefits. So unemployment benefits means like when you finish your PhD, and if you have a gap between your PhD and your next job as a postdoc or something, then you can actually apply for unemployment benefits and you get money every month from the government. So this is also something that is quite good in Germany. So the social security is very good. And the other one is the structured PhD programs. So the kind of program that I was enrolled in, so the Impress graduate schools, where they have a curriculum and they have this orientation programs, and you are supposed to attend certain seminars to get some to finish the PhD. You have some obligations to do, and this is the reason why they also are on par with several English-speaking countries. And then we have, other than the universities, there are several research organizations like the Max-Planck Gesellschaft, which is the Max-Planck Society, and which is covering basic research in the field of medicine, and it could be social science, it could be anything else. But yeah, since we're focusing on life sciences, I would just talk about that here. And then there is a Helmholtz Association. So Max-Planck Gesellschaft is more like the IITs and the IISCs of India, if I can give you an analogy. And the Helmholtz Association is like, it does big research, like which involves huge amount of money, like large scale, like the ISRO and DRDO, that kind of, it's in that scale. And then you have the Leibniz Association, which is comparatively smaller. So it's similar to the IISCRs in India. And still doing very good research. And then you have the Robert Koch Institute, which is like the ICMR. And then you have the Fraunhofer Society, which is more for applied research, for industries, for making patents and so on. And if you want to know more, you can actually go into this link, which is called Research in Germany, and it gives you more insight into the jobs and careers involved for students. And then coming on to Max-Planck Institutes, what is the status of the Max-Planck Institutes in research? If you see the Nature Index, this was 2018, but it remains more or less the same, I think, even now. So you see, it's on par. Max-Planck Society is on par with Harvard, Stanford, and MIT. So it's this is a kind of a rating for the publications that come out and the number of citations and so on. So it's like the research is quite good, quite excellent here in Max-Planck Institutes. So if you want to do PhD, one place to do it is definitely the Impress graduate schools. So the International Max-Planck Schools, and there are like about over 80 Impress schools in Germany, and they are spread across the entire country. Here you see the different states of Germany and where they are spread. And I studied in Freiburg, which is here. And then Impress in biology and medicine alone, we have over 20 different Impress that only focus in life sciences. And okay, if you think that Impress PhD is too much for me, let me just try with masters. then you can try to do masters in Germany, because again, they are world-class master universities in Germany. So if you see the world ranking, the top LMU is a top in Germany-wide is the top most university, followed by Technical University of Munich, and then you have the Heidelberg University, Humboldt University, Charité, and then the University of Freiburg, and so on. So they have also very good world university ranking status. And what is more important is that the kind of curriculum that they have is really very competitive, and it's hands-on. So they give a lot of importance to the practical aspect. So even when you're doing your bachelor degree, you do a bachelor thesis. When you're doing a master's degree, you do a lot of internships, and the thesis sometimes lasts longer than even 6 months. And they have a very holistic approach to study. So they have this combination of biochemistry and biophysics. So these are, I've just listed some kind of MSc courses that you can look for. You can look at their curriculum. So if you don't have the means to study abroad or something, but you still want to be on par with such students, one way to go about doing this is go through their curriculum and see what is so different in their curriculum from yours. And there might be just one or two aspects, and maybe if you can just fill it up with some kind of online courses in India. You can use Udemy or Coursera courses, and try and have as many internships as possible, and try to skill up. And other thing is you have Max-Planck Schools, which is like even more competitive than the Impress graduate schools that I talked about. Here you can do PhD, but you can also do integrated master's PhD program, and it's bigger, and because it has got greater collaborations with a lot of universities in Germany, like high-profile universities. So they also do high-profile research, and then you also have the Göttingen University, which is in collaboration with International Max-Planck Research School. There again, you have this MSc PhD program for molecular cell biology, and they also have another one for neurosciences. There is also MSc PhD, MD PhD. So it depends on how much you want to learn. And so there are so many options in Germany. And you also have the DAD, which is a funding agency through which you can apply. So here is a website wherein you can actually search for different PhD positions available. And this is a funding agency. So if you have a particular if you go into a profile of a particular professor in Germany, and you want to do some research, and you can actually ask this professor if you can have a DAD fellowship, and the professor agrees, he likes your research proposal, for instance, then you both can collaborate together and apply for DAD and get the funding. And the one advantage of this DAD funding is that your house rent allowance is covered, and then you have you also get a free German learning course, which is usually very expensive, but if you get it through DAD, it's free of cost. They cover this up. And then other than this, you have the European Molecular Biology Laboratories, which is like, it's European, so it's not just German, so it's more international. But we have, it is spread all over Europe. And in Germany, you have it in Hamburg and in Heidelberg. And what is in Heidelberg is more interesting to biologists, and you could try there. There is also the program for PhD, which is quite competitive. Okay, so this brings to my summary slide. So why Germany? It is quite high ranking in terms of research, in terms of funding, in terms of infrastructure. And for master students, the best part is that there is no tuition fee here. And so you can, you can work while you learn and try and cover most of your living expenses through that. There are a lot of student assistant jobs available. And then another thing is a surplus funding for research here. You can do cutting edge, high impact research. The work-life balance is very important for the Germans, and you learn a lot from them. Because they really have this 8 to 5 or 9 to 5 job, and afterwards they enjoy their evenings, they enjoy their weekends with family, with friends. So it's like but they at the same time do very good research. So this is something that we can learn from them. And other thing is a social security, as I mentioned, that there is pension scheme, there's an employment scheme. and very good insurance. And the living cost is quite affordable. And another thing is, a lot of parents might think that, how can I send my girl to Germany? I don't know how it's going to be. It's a foreign country, things like that. But I assure you, you don't have to worry so much, because Germany is quite a safe country and it's quite peaceful. Of course, it depends on you, like how you behave, but most of the times it's quite safe. And there have been times when when I come out from lab, it was usually quite late. Sometimes when you're doing an experiment, you forget the time and you continue with that. And then sometimes you see, oh, it's already 11, it's 12, it's 1 in the morning. But then when I go out, initially I was fearful, but it was not that I was fearful of people or something. It's really not like that, and it was super safe. Although I wouldn't recommend anybody to do that, but still, so this is something that you can keep in mind. And then, of course, a PhD is a job, so you earn while you learn, and you also live quite a comfortable life. If not luxury, you can't live a luxurious life, but you live a comfortable life with the PhD salary that you get here. So finally, I would like to thank all the faculty and staff of Dr. M.J. Janaki College of Arts and Science for Women, especially the biochemistry department, Mani ma'am, Victoria ma'am, Sunita ma'am, who was my class teacher, and Manju ma'am. They gave very good foundation for whatever I'm doing right now. And then, of course, my IIT guide, Professor Dr. Pradeep Talapil, and then my PhD guide, Dr. Winfried Romer, and of course the Max-Planck Institute and the Impress Graduate School. And my current supervisor at my postdoc is Dr. Martin Apfelbecker. So right now I'm working on Yersinia, not pestis, but Enterocolitica. And thanks a lot for your time. And I will be open to questions after the next session. Yeah.
Thank you, Ms. Sahaja, for that interesting and highly informative talk. It was very illuminating to our minds. You are definitely an inspiration to our students. Participants, kindly post any questions that you have in the chat box. Now we'll move on to the next speaker. Here is another highly motivated, ambitious alumna, Masai Deepika Akshantala. Masai Deepika completed BSc Biochemistry from Dr. M.J. Janaki College for Women in 2004, after which she pursued a master's in biochemistry from D.G. Vaishnav College, Chennai. In 2008, she completed Masters of Science in Biomedical Sciences from Long Island University, New York. Masai Deepika has more than 12 years of research experience. She began her career as a lab assistant in the Department of Biomedical Sciences, Long Island University. A passion on research assigned many designations in her career. Now she is a senior research associate in Beth Israel Deaconess Medical Center, Harvard Medical School, Boston, USA. A key role and interest include working with medical oncologists, surgeons, radiologists, and clinical research assistants to obtain primary patient tumors and use them for designing and performing co-clinical assays to personalize treatments for cancer patients. Masai Deepika's technical and professional skills include organoids, which is establishing, culturing, and maintaining of tumors derived from patients. A recent technological innovation from Muthuswamy Laboratory, which is used for personalized treatments. She is also proficient in cell biology, immunohistochemistry, animal work, molecular biology, biochemistry, and microscopic techniques. Her research skills are fulfilled with numerous publications in reputed journals. We are very happy and eager to listen to such a self-driven, inspiring personality. Over to you, Ms. Sai Deepika.
Thank you very much, ma'am, for the very kind introduction. Before I get started with my talk, I would like to, from the bottom of my heart, thank all my teachers from the MJC Biochemistry Department, especially Mani ma'am, Victoria ma'am, Sandhya ma'am, Shanti ma'am, for molding the foundation for what I am and where I stand today. It's the way they taught the subject that kindled my passion and my love for this subject, which drove me to pursue higher studies in the field of biochemistry and life sciences. So for the past 12 years, I have been working in cancer research, mainly focusing on breast and pancreatic cancer. And for the last 4 to 5 years, I have been focusing on clinical cancer research. So as you all know, cancer is one of the very deadliest diseases. So almost 9.6 million people all over the world are expected to die from cancer every year. So that's like 1 in 6 deaths is because of cancer. So that's a very high number, especially in developing countries. If you look at the evolution of cancer, this has only been drastically increasing. In spite of so much advancement in technology, research, and medicine, there is still not a cure for cancer, and that's why cancer research is very important. So initially, cancer research were all like one size fits all. So everybody with cancer, no matter what stage, what type of cancer they had, were all given the same type of treatments. But what's more important to know is every cancer is different. Its biology is different. An organ is made up of many types of cells, and cancer from every cell type is different. So if you take the lung, it has small cell lung cancer, non-small cell lung cancer, squamous cell lung cancer. And also, if you take 2 patients having the same type of cancer, they may not have the exact same cancer. And even if 2 patients have the same type of cancer, they would respond differently to the same treatment. A chemo drug may be very effective for one patient, but it may have no effect at all for the second patient. So that's why cancer is considered a very complex disease. So we need something better. So then came the precision medicine, which is genetic based. There are a lot of people around the world doing mutation studies for cancer. Even though it's very informative for doctors to treat, it's not very helpful since the success is seen only in about 10 to 15% of the patient. So we need something even better. So that's why we wanted to take the cell-based testing approach. The reason cell-based testing will work is because it's already been used in microbiology lab. When a patient gets an infection, you grow the bacteria in a dish and test the antibiotics, and whichever antibiotic works, you give it to the patient and the patient gets better. So that's the logic we wanted to apply here. Cell-based testing is by using cell cultures. So this is how a cell looks when they are grown in a flat plastic dish. We call this the 2D cell culture. But the problem here is it does not look like a tumor. It's not representative of the real cell environment, so it's not really predictive of anything. In our lab, we developed a method to grow cells in 3D. You put cells on a matrix bed like matrigel. A matrigel is nothing but a gelatinous protein mixture that resembles the extracellular environment and hence uses it as a basement membrane to grow cells. So you add cells, and then the cells take about 7 to 14 days to form an acini. Acini is like a 3-dimensional structures, which mimics how a cell would grow in our body. So we wanted to study pancreatic cancer using the 3-dimensional model. As you all know, pancreas is a glandular organ. It has a head region and a tail region. So pancreas is made up of endocrine cells and exocrine cells. The endocrine cells secrete the hormones that control our blood glucose levels, and the exocrine cells secrete our digestive enzymes. So the green tube is the pancreatic duct that carries the digestive enzymes from the pancreas to the duodenum and so forth. The cells lining these ducts comprises of the ductal cells and the acinar cells, and when a cancer arises from these exocrine cells, we call it the pancreatic ductal adenocarcinoma, and they comprise about 95% of all pancreatic cancers. So our lab mainly focuses on studying the PDAC. So we resected the tumor, and then we digested it with enzymes to bring it to a single cell suspension, and then we grow it as an organoid in a nutrient-rich media that was developed by our lab. So we call these organoids as mini tumors. So these organoids have all the properties of a tumor, but they're not really tumors, because a tumor will have stromal cells, it has matrix, it has immune cells, but here all we have is tumor cells. Since we knew that it is not a real tumor and it's only a part of a tumor, we asked ourselves, how much can we rely on this organoid for doing anything? We first asked the question, can organoids predict drug response in a mouse? So as with any medical research, we first need to test in an animal model before testing in humans. So if the answer is yes, then we can use this information to go and ask the bigger question, can organoids predict response in patients? So we did a study with 7 sets of matched PDX and PDO models. PDX is patient-derived xenografts. So what we do is we take a tumor cell, we take the tumor cells from a patient after a surgery, and then we inject the tumor cells into a mouse. And we let the tumor to grow in the mouse, and then we do all the studies on the mouse. And simultaneously, we will take the patient tumor cells and grow it in the lab in a dish called in the process of organoids. So that's the patient-derived organoids. So, and then we do a comparative study, and then we were able to show that it matched for all the cancer biology. We were able to test a panel of drugs to see if they responded the same way, if the tumors from the mouse and the tumors from directly on the organoids responded the same way. We did a lot of histology to compare if the histoarchitecture were all the same, and then we did a set of genomics assay to make sure that it was all the same. We have just submitted the manuscript for this, so I'm not able to show any further results. So now that we know that it works in a PDX, we wanted to see how it works in patients. So we made a video here to explain personalized medicine. So when a patient undergoes surgery, they isolate the patient's tumors. So then I will get a piece of this tumor and I will digest it with enzymes to get single cell suspension of tumor cells. I will then grow this in a bed of matrigel with organoid culturing media. So organoids would take about 6 to 16 days to grow into a complete organoid. So from single cells, it has to grow into a bunch of organoids. So we then do an assay to test various drugs. Here you can see that drug B is more effective in killing the tumor cells than drug A. So we can predict that drug B will be more effective for treating that particular patient and create a drug sensitivity report, which the oncologist will then use to treat the patient. So this was our overall plan. And in order to do this, we had to optimize a lot of things. So initially, I started working with surgical samples, but not all patients undergo surgery. In some advanced stages of cancer, surgery is not possible because it's already metastasized to different regions, or if it is too big, it's connected to blood vessels, so surgery may not be an option for the patient. And then we had to work with biopsy samples. This posed more challenges as I had to work with a very small sample size. So it took me at least 6 to 8 months to optimize this entire protocol. So once we knew that this was working, we started doing further testing. So here we know that the models we generate are looking like the patient's tumor. We have done it for many tumors, many patients, but I'll just show you one patient example here. So if you look at the histology of the patient's tumor, and if you look at the organoids, they look the same. So we strongly believe that the organoids mimic a patient's tumor in a lab setting, and that made us more confident that if we do a drug testing on an organoid, it's likely to be relevant for the patient. So with this knowledge, we went on to start a clinical trial at our hospital here in collaboration with some oncologists and surgeons. So this is an overflow of our clinical trial. So we first wrote an IRB to get an approval, and then we set some base eligibility criteria. It involved what kind of patients we have to take in, what stage of the cancer, and if the patient was already treated, if it was a new patient, and all that. So we set up a base eligibility criteria, and then our clinical coordinators team would then go and explain the whole trial process to the patients who we believed were eligible, and then got their written consents for the study. Then our surgeons and radiologists would schedule the procedures for their surgery or biopsy. So in this trial that we just finished, we had consented 76 patients, and I then generated organoids for all these patients. Then my colleague and I, we tested a panel of FDA-approved drugs for different single and multiple drug doses. So then we then generated a drug sensitivity report based on the results we got, and the oncologist used the information that the patient to treat the patient. Unfortunately, since this is unpublished data, I'm not able to show the actual results here. But what are my next steps is we have learned a lot from this phase 1 trial, and using this information, we are now working to start a second trial with a bigger enrollment. Hopefully, once the lockdown is lifted and things return back to normalcy, we would get started. We have done all the groundwork at this point. We've got the approvals and all that. So once the groundwork is done, once the lockdown is lifted, we are looking forward and hoping to start the phase 2 trial for this pancreatic cancer. Then with this experience and with this knowledge, we wanted to see how we can try this organoid models for personalized medicine for different other cancers. So I have also been working on breast cancer on the side. So as we know, breast cancer is the biggest cancer, and it affects women all over the world. And there is very poor prognosis for metastatic breast cancer. So I have spent a few months optimizing different conditions to grow these organoids. Every organ, the growth factors, everything that constitutes in an organ is completely different. So the media constituents, the growth factors that we used for growing pancreatic cancer organoids would not be effective for growing breast cancer organoids. So this needed a different set of growth factors. And also, since there are a lot of hormonal changes involved in breast cancer, it could be triple negative, it could be ER positive, it could be ER-PR positive or triple positive. So based on all this, we wanted to start testing different media conditions. So I've been working on this. I've been working to optimize conditions. I have tried working with PDX models from mouse models, and I've seen some successes. I've seen some challenges that I've been trying to troubleshoot. And then I have tried this on a few cases. We haven't started any clinical trial, but I have tried this with few patients. So here is an example. This is a patient who came to our hospital here, and she was diagnosed with stage 2A triple negative breast cancer. And I was able to get a small piece of her biopsy sample, and I tried optimizing conditions to grow her organoids. And I was able to show that the organoids was able to mimic the primary tumor. And since the patient already had the treatment and this was not a trial, I was just trying to see if I can reproduce the same results. So I tried testing the adriamycin and cyclophosphamide, which the patient already had was treated, and the patient did not have any response. And I was able to try the same drugs to see if I could also see no response. And we were able to match a few things here. But I still need to optimize a lot of things, because what works for triple negative may not work for ER positive. Since hormones are involved, it takes a lot more challenging optimizations to be done. So hopefully I'll be able to get this done, and we are planning to start a clinical trial for breast cancer once we optimize these conditions. With that, I would like to conclude the research part of my talk by saying that I think the future of cancer treatment lies in personalized medicine. This is my team, and every year, this is my wonderful team of oncologists, surgeons, and researchers, and every year we participate in the Pancreatic Cancer Walk to support all the pancreatic cancer patients who are part of our study here. And I have to thank my mentor and my PI, Dr. Senthil Muthuswamy, who has always been a great support and who's encouraged me and who has pushed me to challenge myself and do better in science. And of course, without the support and constant, my parents have always stood by me and always. So I thank them. And with that, I would move on to my career journey. So after finishing BSc Biochemistry in MGR Janaki College, I went to do my MSc in Biochemistry at DG Vaishnav College. That's when I seriously started thinking about future and career and the options. So I grew up seeing my uncle, who is a scientist, who's an ocean microbiologist, and I've always been fascinated with the idea of doing research. Even though I've had like practical classes and we do experiments in the college, I did not know how a research lab environment would look if I would like it enough to pursue my career in that field. So I heard about CLRA labs at that time, and I took a chance, and I went and spoke to a few professors at CLRA asking if I can just shadow them on weekends. It was not part of any project, or it was not an internship or anything. It was just to shadow and to know what a research lab looks, how techniques are done. I mean, we read about all these things in theory, but I wanted to see how everything is done in a lab. So I was very surprised that a lot of professors in CLRI readily accepted, and they introduced me to some researchers in their group, and I was able to shadow them, and I was convinced that this is what I wanted to do. This is the area of field I wanted to take. So then I started looking into doing research or pursuing higher studies in US. So I was advised by a few people I know here that it's better to do. So after your MSc in biochemistry, you can either do a master's or you can go in for a PhD directly. So you need to have 16 years education in India to be able to go in for direct education here in US. So after your MSc, you can directly go in for an MS or you can go in for a PhD. But because I did not have any practical experience, I was advised to take up MS, so I'll have a better idea on the subjects, and then after that pursue for a PhD. So when I started preparing for my master's, the first step was the entrance exams. So you need to clear GRE and Toefl. GRE is if you want to do a Master of Science program, and GMAT if you want to do like an MBA or a management course. So GRE basically tests your English skills and your math skills. And TOEFL is all about English. It's just to see if you can follow American accent, if you can understand, and if you can reply back and all that. So I did take some coaching. So there are a lot of coaching centers. I know of Kaplan, Datamatics, and I'm sure there are many more now. I did my coaching at Kaplan. I only took coaching for GRE. So if you take coaching for GRE, that pretty much covers your coaching for TOEFL as well, because the English side of GRE is what you would need for TOEFL. And something I was advised and that I pass along is the best way to prepare for TOEFL is just watch a lot of American shows. It can be just news. You can go watch American TV shows, or you can just watch documentaries. You can, if you're interested, you can even listen to podcasts, or you can listen to audiobooks. Make sure it's all in American accent. So that's like a good tip to prepare for TOEFL. And then the application process. So how it works is so you can go on the website and look at different colleges. There are a lot of different programs offered by different colleges. So there's like high-level colleges, there's mediocre colleges. My recommendation would be, when you're doing your application process, apply for high-level colleges and also choose a few mediocre colleges. So don't be like, I just want to apply in all high-level colleges. So while you're applying for your thing, the first step you would need is to download an application. The application is actually very expensive. Every college expects you to pay a high fee to just download their application process. So before you go and apply for a college, make sure you've done all your homework on what college you want to do, what courses you want to do, and if you can network and connect with somebody who's already in that college, that would be a great place to start. And start preparing your statement of purpose. So statement of purpose is nothing but you just have to write an essay about how much you are interested in research, or you're interested in your higher studies, how much you're fascinated with biology or biochemistry or what you do. And you can also write about what are your extracurricular activities. It's just for them to say, it's just for you to say that, oh, I'm so much interested and it's my dream to do science or dream to work in science. So it's just like a convincing report or an essay about yourself or your dreams. And of course, you need your GRE and Toefl scores, and you need your transcripts. So a lot of times you may not have finished your final semester exams. So that's perfectly okay while you're submitting your application. If you have your previous semester transcripts, that's good enough to submit. And you can always say that I'm still waiting for my final transcripts, and you can get your application process started. So to every college, you will have to, after you download an application, you will have to attach your GRE scores, your Toefl scores, your transcripts, and your statement of purpose essay and your CV. And see if you can get any hands-on experience, if you have any extracurricular activities, especially if you have done any volunteering. These are some things that they look for. So you can try to add all that into your CV and make it more presentable. So while you're taking your GRE Toefl exams, at the end of your GRE exam, they would ask you they will give you an option to send your GRE test scores free of cost to a few institutes. So make sure that you already have a list of colleges or university that you would want to pick, and then you send in your, you can choose those institutes at your GRE, and then they'll be sending your scores directly to them. And if they like it, they will be able to send you back the records. So, and your student visa. So next step so once you apply and once you get your admission, what you will get is something called an I-20. I-20 is nothing but an admission. It's just a sheet of paper that says you have your admission, you got your admission. So, and then they'll give you all the information on what all you need and all that. So the things that you need for a student visa is, no matter whether you're doing a master's or your PhD, you would need a student visa to study in the US. So for the student visa, you will have to have your I-20. For when you first have to apply for your visa interview, and on your visa interview, you have to make sure that you have your I-20, which is your admission letter. Then you may have to show financial support. So they just need to know that you will be able to pay your tuition fee and all that. One thing is, it's not easy to get scholarship while you're already here in India, while you're already in India, while you're starting with your application process. But once you go there and you get started, so I did not have any scholarship. I did not get any scholarship when I started in India, when I applied for my master's. But once I came here, and if you maintain like more than 95 percentile in your first semester, they will automatically choose you for international scholarship. So the international scholarships are limited for every field, every department. You may have like one scholarship. And you'll be competing against all the international students. So after my first semester, I got the international scholarship, which pretty much covered half of my tuition fee for the rest of my 3 semesters. And you will also be allowed to work on campus here. So you can keep all that in mind. So don't be disappointed if you do not have a scholarship. You can always work on it once you come here. So you will have to show some student visa. You'll have to show at least some loan. You'll have to show that you are applying for a loan. You have to show that you have applied for a loan and you have a proof that your loan is approved while applying for the student visa. And then at your interview, you may be asked the same questions as what you I've said in the statement of purpose. Why are you so interested in pursuing your science? Why do you think this is your passion? So just go confidently to their visa interview and answer all these questions, and that should be it. So opportunities. Once you finish your, so you can either come in for master's or PhD, or you can finish your master's and then apply for your PhD. And that was my idea. I wanted to finish my MSc, and then I was preparing to get started with my PhD. And due to personal reasons, I couldn't pursue my PhD. But you still have a lot of avenues. You still have a lot of openings and opportunities with masters here. So if you want to pursue in research, there are two options. You can either go with academics or industry. So academics is when you are part of an institute or an university research, and industry would be like industrial companies, like in India, if you had to say Biocon and all that, or here Johnson & Johnson and Novartis and all that. So there is pros and cons to be working on academics and industry. Academics, you have your complete freedom to pursue your research. You can choose your own topic. You can just work on anything you like. You can just start exploring and asking your own questions. But whereas in industry, that is very limited. So you are expected to work based on the questions the industry is interested in asking. And when you look at your pay, academics would pay way less compared to industry. You'll get paid very high in industry, but whereas in academics, since everything is grant-based, it depends on how much, if you have your grants, if you have your fundings, depending on how much funding your PI has. So that limits everything in academy. But in industry, you have like a set salary, and it just keeps increasing. And also in academics, there is no fixed time. Your experiments decide your time, your days. Sometimes you have to work 7 days a week if you're doing some experiments. Some days it's just 3 days a week. Some days you're always in the lab. But whereas in industry, it's more like a 9-to-5 job. You decide what you have to do today, and you can go on with it. So you can choose it based on what you are looking for, but you also have options of, you can start with academics and then you can go on and move to industry, or vice versa. So the transition here is very easy. So are the transitions with different branches of life sciences. So you can start with one, and you can get your hands-on experience. You can take on some trainings. There are a lot of online courses available here that you can keep taking to better yourself. And then once you think you're proficient in something else, you can very easily try your hands in a different field. So I started with cell biology, and I majored in immunology and cell biology, but now I'm not working anything related to immunology. I'm pursuing in cancer research. There is very, very high scope for bioinformatics. So almost all clinical trials group, we have to coordinate with bioinformaticians here to get our data sorted, and they play a very, very important role in all our publications, in all our studies. So if you're interested in pursuing in bioinformatics, there is a very wide scope for that in US. And another option is clinical study coordinator, where you will be the study coordinator for any clinical trials. You'll be the one you'll be the middle person between the clinic and the research lab, and they play a very important role in making sure all the clinical trials go on well. And it's a very interesting and fascinating job that has a very high scope too. And of course, I covered PhD and science writer. If you have a passion for writing, and if you have a passion for science, you have so many opportunities here. You can help with grant writing. You can work in a lot of journals like Nature, Cell, and Cancer Research, and all these journals. You can work as an editor, and you can you can start with writing for small articles and all that. So the opportunities are vast here. And once you have a dream, you can just keep pursuing and you can just keep going on. With that, I will stop and I'll take questions. Thank you. And I would like to thank MJC Biochemistry Department and all the departments for giving me this wonderful opportunity. Thank you.
Thank you, Masai Deepika, for the highly informative and factual speech. Your career journey is very motivating to us, and I'm sure our audience had a satisfactory information for their thirst. And now we'll move on to the question and answer session. The first question is to Ms. Sahaja. This is from the ID Gopi Shivapriya. How Western blotting involves in nanoparticle drug delivery? Can you mention about the mechanism?
Okay, it's so in Western blots, what we actually see is that if you're looking at a particular signaling pathway, you want to see if something is affecting a particular signaling pathway, then you can use and for example, if your nanoparticle is known to affect PI3 ACT signaling pathway, I'm just giving you an example, then you want to see what is the differential expression, so the protein expression levels, and that's where you will use Western blotting. So you can take your nanoparticle, treat it with the cells, prepare lysates and load, and see with different with different nanoparticle concentrations or different size of the nanoparticle, what is the effect. So this could be one idea. Yeah.
Okay. Thank you, Ms. Sahaja. The next question is to Ms. Masai Deepika. This is from Kavya Murugesh. What are the ways to treat the types and stages of cancer? I think she's meaning about the organoids. How would you treat the types and stages of cancer?
So the organoids, we do a Muthuswami method, and there are so many other methods for treating cancer using organoids, and there are many methods to teach to grow organoids.
Okay.
We usually test it for final stage cancers,
Okay.
and it can easily be tested for. Currently, there are organoid treatments for breast cancer, pancreatic cancer, lung cancer. Lung cancer is being a challenge, but there are teams working on it. Prostate cancer, and there are also groups working on brain cancer.
Okay. So the next question is to Ms. Sahaja. This is from Gayatri Devi. She wants to know if you can suggest some simple methods or techniques to evaluate the genetic expression of test cell lines that have been treated with our drug and control cell lines.
Okay, so I have never done such a study during my, I've never done it. But then I could think that about the dosage dependence, and if you know like which pathway your drug is going to affect, then you have some, and also you have some control genes which your drug won't affect. Then you can do a gene profiling, you can do RT-PCR, and I don't know too much about this field, but maybe that could be a good start to know, first of all, what is your cytological effect. So if it's affecting apoptosis, or which is the pathway that is where your drug is supposed to act, and then move on with gene profiling assays. Yeah.
Okay. This question is to Sai Deepika. This is from Dr. Durga. What are the ethical challenges that are related to organoid technology?
So yes, there are a bunch of ethical challenges because everything is IRB based. We have to make sure that we get complete clearance. We are completely transparent on what we are doing, how much, what we are using their samples for. We have to say that we are going to use the organoids for testing this and this and that, and we are going to use your blood samples for testing this. And it's all on paper, and we cannot use the samples for doing anything else other than that. We cannot use it for our research. We cannot ask any questions or do anything. That's that's not part of the concern. So that is a challenge. And so with the phase 2 trial, we are trying to see to accommodate more questions so we can widen our scope of research there as well. And also with the patients, there's always a stigma there that about cancer, right? So they're very concerned about having them part of the clinical trial. A lot of times the patients are like, oh, we don't want to do anything. We don't want any of this. We don't want to get involved in any of this. And we just give us a treatment and we will go. But there are also like last stage cancer patients who are like desperate, who are like, we are willing to try anything. Just even if it will buy us more time, we are willing to try anything. So we do face a lot of ethical challenges here, and we are trying our best. That's where our clinical coordinators team comes in, and they try to bridge the gap. They try to make the patient very comfortable. We keep the patient informed all the time. We keep giving them updates, weekly updates or monthly updates on saying what's happening, how are we doing, and sometimes these patients, they are curious. They want to come to the lab and they are like, can we come to the research lab and see how our organoids look, what you're doing with it? Can we see in the microscope? And we usually are okay with that. We get them to our lab. There are a few patients who have come to our lab and who have seen their own cells growing in a dish, and it gives them hope. So yes, so there are ethical challenges, and we are trying our best to go past that.
Okay. Owing to time, we'll just take up another few questions. This one is to Ms. Sahaja. Is doing a course in German language mandatory before enrolling in a PhD program?
No, it's not. But then it would be nice to know the language. But since the coursework is completely in English, you don't really need to know German before coming here, for PhD especially. For master's program, it's a different scenario. There are several master programs which are in English, and some are even bilingual. So then in that case, it's good to have a good, at least a little knowledge of German. Yeah.
Okay, thank you. And this question is from Akshaya to Ms. Deepika. Does CRISPR gene editing techniques, will it be useful in treatment of cancer?
It is. So there is precision medicine. I'm not an expert in answering this, unfortunately. I'm sorry. But yeah, I know a lot of teams are working on CRISPR. We are not focusing on it in our lab currently, but there are a lot of people working on CRISPR genes for precision medicine now.
Oh, okay. This question is from Madhumita. This is to Ms. Sahaja. What are the job opportunities for MSc Biotechnology students there?
So a lot of students get absorbed into pharma companies. They into quality as quality analysts. So quality control, and then some also go into technical writing and totally different fields. But usually it's in pharma companies. Yeah.
Okay. Ms. Deepika, there's one question.
Yes,
how are organoids being successful in mimicking the immune system?
Oh, yes. So we have an entire team now in our lab working on immunology. We have some experts working on it. And so we are trying to get the immune cells extract the immune cells from the blood cells, from the blood of the cancer patient. And then we are trying to isolate these T cells and grow them with the organoids to see how they are working. And we are seeing some success here, but it's still work under progress.
Ms. Sakaja, Dr. Manimegle has asked you to brief on DAD fellowship. I think you had mentioned it in your PPT. Just some key points on it, please.
So that fellowship is usually for either you can apply for masters or majorly for PhD programs. So in this, you get 1 year. I think initially you get 1 year scholarship. Actually, I don't know entirely about this fellowship. I can only give you a few overview about this, a little overview. So it's like And so you usually have to have a research proposal, so which is like 10 to 12 pages of your research that you would like to do. And best is to have some PI who would like to take you in his or her lab for doing the research. So once you have that, and you have to apply online, they have deadlines for it, and there are a lot of information for different kinds of scholarships coming within the umbrella of DAD fellowship. So it is in the website, and so basically it's better that you have a PI who has agreed to work with you, and you have a very good research proposal. So yeah, that's the thing. And then they also help you in learning German, so you can apply for German courses. So it's in a way, I think they would like, although the PhD would be in English, since you're coming to Germany, I think they like, since they're also a German fellowship, so they funding agency, they would prefer that you somehow learn German. It's like promoting their language. So that's an added advantage, that you get 3 months to do just that, and then go on with your research once you come here. And they provide also the house rent allowance and scholarship for a year to begin with. Yeah.
Okay, thank you for that. Ms. Deepika, one last question. Can genetic engineering be applied to the organoids apart from the assays?
I think so, but we are not doing much. We are analyzing all these samples for genome sequencing. We get the blood from the patient and we analyze we extract DNA and we analyze the genome of the patient, and then we do the same testing. So we have a wonderful bioinformatician in our team, and she does all this work of extracting DNA from blood cells and matching the sequence with the organoids. So that's where we are at, and we haven't done any genetic engineering, at least not in our lab.
Okay. Thank you, Ms. Sahaja and Masai Deepika, for enlightening our participants. Thank you for your patience and time. I now request Dr. Nitya Kalyani, Assistant Professor, Department of Microbiology, to present the repertoire of these alumni lecture series.
Thank you, Deepa ma'am. Good evening to all. Dr. M.G.R. Janaki College of Arts and Science for Women has many illustrious alumni who have made us proud time and again. Alumni of the departments of microbiology and biochemistry has distinguished themselves through achievement and professional accomplishments. As an alumna of the Department of Microbiology, it gives me immense pleasure to present the report of Alumni Lecture Series 2020, a series of open lecture by MJC alumni on opportunities in biosciences. MJC's Alumni Lecture Series, featuring alumni from the departments of biochemistry, microbiology, and PG departments of applied microbiology, bioinformatics, and clinical trial management, to share their will and wisdom with students' community and help students think about the transition after graduation. Engaging topics, thought-provoking discussions, and networking with alumni and students' community are the success elements of this alumni series. I'm sure the alumni lecture series provided an insight about the career paths our alumni had chosen that led them to where they stand today and the varied opportunities in biosciences. The first speaker of the lecture series was an alumna of the Department of Microbiology, Ms. Deepa, a research scholar in Cancer Institute, Chennai. In her lecture, Ms. Deepa highlighted the antimicrobial effect of probiotic ECN, potent enough to maintain the viability of cells during gastroenteric infections, and how ECN protects the host system from the pathogenic stresses and significantly reduces such infections. She also encouraged the students to keep exploring new avenues that would help them grow. On day 2, an alumna of the Department of Biochemistry, Ms. Ramani Sundaram, who is currently the team lead, Research, Training and Development, Nightingale's Medical Trust, Bangalore, shared her views on avenues in socio-medical care with relevance to dementia, the pharmacological and non-pharmacological interventions in dementia pathology, which may offer the benefit of improving quality of life by reducing cognitive decline and incident dementia. She highlighted the uniqueness of biomedical research and how the findings of the biomedical scientists are instrumental in making the advancements of modern medicine. Our next speaker, Dr. Sukanya Shamsundar, Research Fellow, National University of Singapore, and old NJCN of the Department of Microbiology, gave insights on the career opportunities in the field of biosciences. Talking about her transition from a student to a postdoctoral researcher, she shared how she started her career as an intern at IIT and waded through the waters of research till she found the shore. She also motivated the students to set smart goals and to be consistent in professional life to become more successful. Day 4 witnessed a knowledgeable session on precision medicine, also known as personalized medicine, a new frontier for healthcare combining genomics, big data analytics, and population health. Ms. Vishnu Priya, Associate Researcher, National Institute of Research in Tuberculosis, Chennai, and alumna of the Department of Biochemistry, threw light on non-conventional opportunities in biosciences. She also described the progress, challenges of precision medicine, the futuristic capability of NGS techniques and CRISPRs as essential tools of precision care, and the exploration of biomarkers as the main branches of precision medicine. She also discussed the reliable detection prospects of nanotheranostics for diagnostics of diseases at an initial stage for optimum treatment. On the fifth day of the Alumni Lecture Series, an alumna of the Microbiology Department, Ms. Deepti Pavani, research student, Belfast Metropolitan College, UK, aspired the students to study and work in UK owing to its high career prospects and excellent lifestyle options. Another alumna of the microbiology department, Ms. Chatrupa Mukherjee, engineer, technical communication, Siemens Healthineers, Bangalore, spoke about the alternative, prominent, high-paying career options available outside academia and industry for life science graduates. She emphasized on downplaying the specifics of one's degree and focusing on broader interests and abilities to look for further challenges. Some career options listed include a startup entrepreneur who solves relevant problems throughout off-the-box innovations, technical support specialist in a company to complement the sales and marketing departments, project manager in government or private program, intellectual property, technology transfer, data analytics, medical coding, medical billing, and scientific research writing. Know thyself. This old adage was endorsed by both the speakers. And today, prominent alumni of the Department of Biochemistry, Ms. Sahaja, postdoctoral researcher, Institute of Medical Microbiology, Virology and Hygiene, University Clinic, Hamburg, Germany, and Ms. Masai Deepika Akshantala, senior research associate, Harvard Medical School, Boston, USA, imparted wisdom that will inspire the future generation. German universities have an outstanding reputation throughout the world. Each year, they produce significant internationally recognized advances and innovations. In her talk, Ms. Sahaja provided the necessary information a student needs to get his or her life started in Germany. She also briefed on the excellent research opportunities for international students and researchers in Germany. In that same spirit, Ms. Sai Deepika expressed agreement on the growing role of precision to personalized medicine for the treatment of cancer, which shifts the cancer paradigm to prevention rather than reaction. Our heartfelt thanks to all our alumni for motivating our students to search, create, engage, explore, and to excel in what they do. I'm sure this initiative by the Departments of Biochemistry and Microbiology, relevant to the entire student population, has been a great success with the students. I'm greatly indebted to our correspondent, Dr. Lata Rajendran, Chairman Mr. Kumar Rajendran, for giving us the opportunity to organize this alumni lecture series. I'm thankful to the dynamic ladies of our college, Principal Dr. Mani Meglai, Vice Principal Dr. Lakshmi Balaji, Dean Students Dr. Apita Sabapati, and Dean Academics Dr. Shanti Lakshmi for their unstinted support and guidance. A special note of thanks to our Principal Dr. Mani Meglai for sparing her time to lay down the foundation and to provide the context for deliberations during the 6 days of the webinar. Advancement in technology has brought a paradigm shift in every aspect of our lives, especially during this lockdown. One key shift is replacement of seminars by webinars. Transitioning a traditional seminar to a virtual environment Delivering the content through a webinar was initiated by the faculty of the Departments of Biochemistry and Microbiology. I acknowledge the unwavering support received from the faculty of both the departments, specifically for their incredible job in organizing the webinar, managing the website, and providing the technical support. In addition, I acknowledge with gratitude the support provided by Dr. Uma Shankari, Shifter Coordinator of a college, and Dr. Sumati, Assistant Professor, Department of Computer Application. It has been a real pleasure to work with these very fine and capable ladies. Finally, let me thank the participants who have responded so well to our invitation to participate in the webinar. Dear graduates, always remember that a college degree is an indication that you are prepared for life, and achieving excellence is never an accident. It is a result of high intentions, sincere efforts, skillful execution, and ability to see obstacles as opportunities always. Best of luck and congratulations to all. I once again thank everyone whose contribution has made this webinar a successful one. Every ending is always a new beginning. When something ends, something new begins, and a new beginning is always a new opportunity. As a means of providing opportunities to students to grapple with specific information on the trinity of COVID, immunity, implications, and interventions, the Departments of Microbiology, Biochemistry, and PG Department of Applied Microbiology and Bioinformatics and Clinical Trial Management is organizing an international webinar series on 4th, 5th, and 6th May 2020 between 5 to 6 p.m. I request the faculty and students to register for the webinar. Hope to connect with you all in the next webinar series. Thank you.